Our next presentation is Catalent Pharma Solutions, and I'd like to welcome Fred Cedrone, Vice President, Corporate Innovation, to the stage.
Thank you. Well, hello, good afternoon, everyone. I'm going to give you a brief overview of Catalent cell and gene therapy, and we will dig deeper in the iPSC field. I will show you ways to access technologies and to build partnerships that can ease going faster to the clinic. So, you know that Catalent is a global CDMO, serving partners across all modalities, including cell and gene therapy, and we will focus here. Catalent can be supporting programs from very early development to commercial stage. We can accelerate and de-risk the development of your treatment by bringing the expertise and the know-how that we have acquired across decades of GMP manufacturing in relation with the FDA. We have assisted nearly 50% of FDA approval last 10 years. What is less known is that Catalent does not work only with big names. In fact, 80% of all cell therapy partners are small biotechs. With small companies, we are acting as enablers, bringing technologies and ready-to-go solutions that can help going faster to the clinic. And we build partnerships that allow early biotech onboarding Catalent and technologies at a very early stage. Let's have a quick look to the cell and gene therapy network. Catalent manufactures plasmids, viral vectors, mRNA, protein, iPSCs, and cell therapies. By partnering with Catalent, you will access a global network of capabilities, expertise, and sites that are all interconnected. We can build fully integrated solutions from accessing the starting material like off-the-shelf plasmids, off-the-shelf GMP iPSC lines, to the final formulation of the drug and drug product release. For example, we can use Catalent plasmids that have been fully optimized as starting material for AAVs or manufacture yours, and then ensure the process development and manufacture of your AAV gene therapy. Same. Plasmids can be used for lenti. The lenti can be used to transduce the cells, and then we can manufacture the CAR T cell, QC them, and release the drug product. GMP iPSC as well can be the starting material that we provide, or the iPSC can be coming from the reprogramming of the specific cell type the partner wishes to use. Then we can add a layer of gene editing, for which plasmid, mRNA, protein can be used to deliver the functional enzyme, but also AAVs and plasmid that can bring the repair matrix for knocking in. All of these pieces will be manufactured with the required quality that will ease the downstream regulatory discussions. Just talking about iPSC, I can show you the network. So Catalent Düsseldorf is the iPSC Center of Excellence, where the iPSC lines, the differentiation protocols, the expansion, and all the required technologies are developed. I like to say this is where the magic occurs. Düsseldorf site is equipped with GMP suite, so the process development, analytical development, and GMP manufacturing can occur in Düsseldorf, but for small scales. Then we have Gosselies in Belgium or Princeton in New Jersey. These two sites are our cell therapy development manufacturing site for large scale. Each site can do process development, optimization, scale-up, analytical development, method qualification validation, GMP manufacturing, QC, and drug product release. The three sites are acting as one network across the three locations. For example, Düsseldorf can develop iPSCs, bank the lines, and support R&D using GMP-friendly conditions. Then our partner can select the manufacturing site depending on its strategy, US, Europe. The three sites are operating under the same quality umbrella, same quality system, same QP. Looking ahead, when we'll be reaching the late stage, Gosselies and Princeton can be the secondary site or backup of each other, meaning that you can de-risk the program itself. Especially interesting when you are going to the commercial stage. In terms of iPSC workflow, Catalent can support across 4 facets, providing off-the-shelf GMP iPSC line under our license that are equipped for going to cell therapy development, but also R&D line and differentiated cells for testing. We can use developed protocol for reprogramming and for differentiation to the final lineage. The protocols are covered by IP, which is good for the end product. We can be performing gene editing service, but also answering banking and process development, scale-up, and GMP manufacturing of the iPSC-derived cell therapy. There are many entry points for our partners. The partner can bring the iPSC line, the differentiation process, and then Catalent adds the missing pieces. But in sometimes, some cases, the partner just comes to us with an ID, a proof of concept at the research scale, and we bring all the other pieces. The off-the-shelf iPSC lines are GMP from day one. They are from neonatal origin, cord blood, which means a very low mutation burden. They have undergone an extensive QC for reprogramming vector clearance, morphology, gene expression, karyotype, and differentiation potential. The recent FDA guidance is stating a whole genome sequencing with a 50-fold coverage. This is what we were doing already 5 years ago. Regarding the lines themselves, they are from European or US origins. The European lines are already with the FDA. The mitigation has been performed, and one line is already accepted to be used in the US. The US lines are fully FDA compliant, and all the lines have been tested for differentiation potential, either by internal testing, either by our partners. The lines of the shelf mean the donor eligibility requirements have been reconsented for commercial exploitation and are available through a license. In addition to the off-the-shelf GMP lines, we can bring patented protocols that have been developed for scalability and GMP compatibility. We can access immune cells, differentiation and expansion, gene editing, for which the team is able to reach a very efficiency, knocking in without any selection marker. We can do RPE cells, highly pure and cryopreservable. We can do cardiac cells, highly pure, with a very robust and scalable process, and MSCs as well, with a huge expansion potential. And I'm just going to give you an example of an iPSC-derived platform for immunotherapies that we can build. So starting from an off-the-shelf GMP line and applying a succession of media, the iPSCs are expanded and turned into hematopoietic precursor cells, leading to 100% pure CD34 plus cells, with a very high purity and a very potent cell, and very potent cells. Then, the hematopoietic precursor cells are differentiated into NK precursor cells. These NK precursor cells are cryopreservable. We all know that NKs are super sensitive to stress like cryo. So our team has developed a method enabling to harvest and cryo NK precursor cells at a 90% recovery rate. These NK precursor cells are then differentiated into NK by applying succession of media without using any feeder cell. That we all know can be an issue. Finally, we get NKs with a high killing potency. 70% of targeted cells are killed with a 1-on-1 ratio after 4 hours. And the process has been further optimized to be compatible with multiple cell lines. Then we can consider that we need to apply gene editing. Knocking out genes are not that an issue, even if it can be quite difficult when we are starting to do multiplexings. But targeted knock-ins without any enhancer, without any selection marker, especially in view of doing GMP manufacturing, is not easy. We are all aware that we commonly reach 2 to 3%, which is quite low. So by applying a series of optimization, the team is reaching 40% targeting knock-in efficiency using GMP-friendly conditions. This is iPSC line independent, target loci independent, and CRISPR independent. The edited cells are functional. There is no interference with the NK differentiation, and there is no interference with cardiomyocytes. As a summary on this slide, we do see that we can build a fully integrated NK platform starting from off-the-shelf GMP iPSC lines, using a GMP-friendly protocol for gene editing, then a GMP-friendly NK manufacturing process based on an iPSC expansion, NK precursor differentiation, cryopreservation, and final feeder-free NK expansion. And we can scale this up to a commercial stage. So briefly, by partnering with Catalent, you can access expertise, resource, and capabilities for the development and GMP manufacturing of your product from very early stage to late stage and commercial. The development and GMP manufacturing are done under standard service agreement. But in addition to this, Catalent is developing and acquiring technologies with the goal of helping its partner going faster to the clinic by providing the right tool, the right technology, and resource at the right time. You can get off-the-shelf GMP iPSCs under a license, but also their R&D equivalent and R&D differentiated cells under an MTA. This is useful to get the POC. But also you can get differentiation protocol, expansion, and gene editing methods that are available under licenses. The key point is that we will build partnership and licensing models that are taking into consideration the development stage of our partner. True, a biotech is not a pharma. I'm coming from the biotech world. I know very well the value of cash. So knowing the funding environment is very difficult, we will work together to target the main inflection point the VC have in mind. Is the manufacturing platform ready? When are you going to file your IND? When are you going to dose the first patient? And when are you going to get the first clinical data? And we will develop a model that will reflect your strategy and timelines. For a biotech, focusing resource on process development and getting the product ready to go to clinic is key. Happy to take any question. Thank you.